Hello, my name is John, and I'll be talking about the relationship between cholinergic and noradrenergic activity and behavioral state in mice. This poster depicts my current work towards my thesis project and partial fulfillment of the requirements of the Robert D. Clark Honors College. An animal's ability to engage with a particular task is largely variable. Oftentimes, they are engaged and focused, while other times they are quite distracted. However, it has been previously shown that there is an optimal arousal state under which an animal can perform optimally. In short, arousal state describes an animal's engagement with the environment and surrounding external stimuli. Arousal encompasses both brain state, which describes the physiological activity of the brain, and behavioral state, which describes observable behavioral patterns and movements of the animal. Moment to moment changes in brain state and arousal ultimately influence an animal's ability to successfully perform various perceptual or cognitive behavioral tasks. It is possible to investigate the relationship between these changes in behavior and their underlying neural circuitry through a careful analysis of external behavioral motifs. The second set of figures illustrates a common behavioral assay in which we can acquire various behavioral data, including walk velocity, whisker motion, snout motion, and pupil diameter. Previous literature indicates that both walk velocity and pupil diameter are closely related to changes in memory potential of cortical neurons. We can therefore utilize these external behavioral motifs, such as pupil diameter, to partially explain these moment-to-moment fluctuations in behavioral and arousal state and their underlying neural circuitry. Rapid variation in behavioral state is, in part, influenced by various neuromodulatory systems within the brain. Neuromodulators are neurons that modulate the activity of other neurons. Varying activity within these neuromodulatory systems produce changes in an animal's cognition and sensory processing by projecting to various parts of the brain. Two predominant neuromodulatory cell types, cholinergic and noradrenergic neurons, produce and release the neurotransmitters acetylcholine and noradrenaline respectively. Cholinergic projections from nuclei in the basal forebrain and noradrenergic projections from nuclei in the locus coeruleus innervate to various target regions in the cortex, ultimately influencing cognition and sensory processing. These changes in neuromodulatory activity are linked to changes in arousal state and behavioral state. And so, this background forms the basis upon which I will approach my project. My project will investigate this relationship between cholinergic and noradrenergic activity and behavioral state in three ways. 1. Confirm the relationship between neuromodulatory activity and arousal. 2. Determine whether changes in neuromodulatory activity precede or follow the onset of behavioral events. And 3. Determine if acetylcholine and noradrenaline neuromodulation across multiple cortical regions is temporally related. To accomplish these tasks, we must first visualize cholinergic and noradrenergic axon activity in awake mice. The activity of these noradrenergic and cholinergic axons were visualized in mice injected with the GCAM6 virus. DBH-Cree mice were injected in the locus coeruleus, and Chat-Cree mice were injected in the basal forebrain with GCAM6 virus, a fluorescent calcium indicator that allows cholinergic and noradrenergic neurons to fluoresce under microscopy when active. This virus will then project from these nuclei to the surface of the cortex, allowing us to visualize their fluorescence. After mice are injected with the virus, we conduct simultaneous 2-photon axon imaging and behavioral data acquisition to directly compare axonal activity to observable behavioral motifs. We can collect metrics of behavioral state, including pupil diameter, snout motion, whisker pad motion energy, grooming, tail motion, and walk velocity. We will also conduct histological verification of injection sites to confirm that we are actually recording from either cholinergic or noradrenergic axons. My preliminary data indicates that after performing histological verification of injections into either locus coeruleus or basal forebrain, we can successfully observe fluorescence in cholinergic and noradrenergic axons. Additionally, after performing 2-photon microscopy while concurrently recording behavioral variables such as pupil diameter, whisker motion energy, and snout movement, we found that changes in behavioral state are related to observable changes in neuromodulatory activity in the cortex. Delta fluorescence over baseline fluorescence, or DF over F, for cholinergic axons increases as an animal becomes more aroused, or as we see variation in behavioral state. These data support previously cited literature stating that changes in cholinergic neuromodulatory activity reflect changes in behavioral state. Future work on my project includes analyzing temporally how neuromodulatory activity is related to the onset of behavioral events. Additionally, in order to further our understanding of the acetylcholine noradrenaline neuromodulatory systems within the brain, we'll determine if the neuromodulation of multiple cortical regions is temporally related. Thank you very much for listening.